Thank you, Susie. So I'll be sharing with you today some of our work, as Susie mentioned, trying to use arterial spin labeling on the Connectome scanner to look at cortical depth dependence of these perfusion signals. So just a brief overview of ASL. So the contrast in ASL is introduced through, it's introduced through radiofrequency inversion of the spins in the arterial blood at the level of the cervical spine. So if you look, there we go. So the region highlighted in red here are shown overlaid on top of T1 images acquired and positioned as if the individual was in the head first supine position placed in the scanner with arterial flow going from left to right. So the red region here is where those RF pulses are played out to actually invert the spins of the inflowing blood. And then we wait a little bit of time, typically 1 to 2 seconds, known as the post-labeling delay, to allow that blood to flow from the neck into the brain, at which point we acquire 2 sets of images in the whole brain, one in this label condition and one without the label condition, known as the label and control images. And then a subtraction of these control and label images gives us a perfusion-weighted signal, which is then fit into a kinetic model to arrive at CBF maps and quantitative units. So we and others have used ASL at 3T to look at how perfusion in the gray matter cortex and in the white matter change as a function of age. So what these maps are showing on the top are surface-based renderings of statistical tests looking at CBF as a function of age in the cortical gray matter. And every region you see in the blue or the light blue here are regions where there's a significant association with lower CBF and older age. And the same is true with the white matter as well. So if we apply these transformations to look at CBF as a function of the white matter skeleton, we see a pretty widespread association between age, older age, and lower CBF. But what we know generally less about is how that CBF, that perfusion value, changes as a function of cortical depth. And one of the limitations of ASL that makes this a really hard question to answer is that the acquisitions tend to be pretty coarse in resolution. So if we look in the plane direction, it's generally between 3 to 4 millimeters and about 4 to 8 millimeters in the through plane direction. So you can see on the right, even with 3D acquisitions, that the resolution makes it quite difficult to resolve the gray matter from the CSF and white matter, much less within the cortical ribbon. In addition to that, what we also see with 3D acquisitions in ASL is the through plane blurring. So you can see in the coronal and sagittal slices of these acquisitions, there's significant blurring in the Z direction that makes it really difficult to resolve these structures. So pretty recent work from Danny Wong's group at USC has shown the feasibility of being able to do this at high field. So these are 7 Tesla results on the left using 3D GRACE in a limited field of view application, showing gray matter perfusion as a function of cortical depth here, going from the white matter surface to the peel surface from left to right. First in the motor cortex, and these orange plots here are the ones where the data was acquired in the resting state, and also in the visual cortex on the right. And in both cases, what we see is that the perfusion tends to increase as you go from the white matter to the peel surface up until a certain point, where it seems to drop off as you get to that peel surface. The graph on the right is much more recent data that was published this year, looking at using the 7T impulse scanner to be able to look at these same data, going again from white matter to peel surface on the left or right. And what these are showing is a pretty good correlation between the CBF values across the cortical depth with cell body density imaging data that was acquired independently of that. So this shows really the promise of being able to use ASL with the right methods to be able to look at this. So the question that we had is, can we acquire similar high resolution whole brain perfusion imaging data at 3T with standard sequences to be able to look at this as well? And here is the list of parameters that we use for our sequence. That the main things to keep in mind are the labeling duration and the post-labeling delay were able to be implemented as typically recommended at 1800 milliseconds each. And we're able to push the spatial resolution down to about 1.8 millimeters in plane and 1 millimeters in the Z direction. And the total acquisition time was relatively long at 18 minutes, but we were able to acquire 44 control label pairs to be able to average over a long number of dynamics there. Here are just a couple of exemplary images that were acquired in a 40-year-old male subject, I believe. And these are control images showing in all 3 orientations that we can acquire these data at this high resolution without any significant artifacts being evident. And these are perfusion-weighted images acquired and processed in that same individual, so showing 4 different axial slices on the top, where we can see the expected gray matter to white matter contrast in these images, and also the variability that's apparently present in the cortical blood flow across different thicknesses, and also in different regions of the gray matter as well. And importantly, on the bottom, we can see in the sagittal and coronal orientations that the through plane blurring that I showed in the typical acquisitions is largely absent, and we can acquire these data at this resolution without introducing those blurring artifacts in those directions. If we take that CBF data and project it onto the cortical surface and look as a function of a cortical depth, so you can see here on the left a projection on the white matter surface, and on the right, the peel surface, and then halfway in between here. And qualitatively, the regions pointed out by the white arrows here show regions where we can visually tell that there's a difference between these perfusion values across the different cortical depths. And so using these promising findings, we then enrolled a larger number of individuals. So we looked at 2 different age groups to look at the effect of age on these different quantities. And we had 2 different groups, one ranging from 22 to 26 years of age, and the next from 70 to 77 years of age. So it was pretty distinct groups to try to narrow down that age effect. And if we look at 2 different individuals in these groups, so one is a 26-year-old female on the top row there, and then a 74-year-old on the bottom row, we compared 3 different ASL acquisitions in all of these individuals. So the first column here is a conventional 2D EPI acquisition at conventional resolution, so around 3.5 millimeter isotropic. That next row here shows a 3D Grace acquisition with a similar resolution of about 3.5 millimeters. And that last row here is the higher resolution 3D Grace that we mentioned. And these arrows are pointing out regions where we can distinguish visually CSF space from gray matter space in the 3D Grace. That's the high resolution that we can't really make out with the lower resolution implementations. Lastly, we've done some pretty preliminary quantitative analysis to try to look at this depth dependence effect. And so we transformed these CBF maps into FS average space using FreeSurfer, calculated average CBF values inside of regions of interest using the DK atlas, and then looked at the effect of layer and age on these values. So first, so I'll apologize, these color bars didn't reproduce exactly how I was hoping, but essentially what these images are showing on the left are the effect sizes, where every region that's in red or yellow shows a gradient going from the white matter surface to the peel surface, where the white matter value is lower than the value on the other side. And the statistical maps here are showing the regions where that comparison is significant. So this is including all individuals in our study, so both the younger and the older cohort. But if we look at the interaction effect and try to look at the effect of age, what these images are showing are everywhere where there's either blue or light blue are regions where that gradient from the white matter surface to the peel surface is attenuated in older individuals. So this is a pretty widespread effect, as you can see on the statistical maps on the right. And if we narrow down and look at these individual trajectories, what we're seeing is that the young individuals on the right here show that increase from the white to the peel surface that we see. But in the older individuals, it looks like that effect is attenuated. And what this potentially tells us is that this effect of age on the cortical perfusion may not be uniformly distributed across the cortical depth. While this is representative, this insula case on the left is representative of the group average results, there are regions that we found where the patterns look quite distinct from this overall group level effect. So you can see in the parahippocampal gyrus on the right that the patterns on the individual level and also as a function of age are quite distinct. So it's we believe it's going to be important to look at these results in larger cohorts as a function of these different regions to see how consistent these relationships are across the cortex. And so just to conclude, we believe that these data show the promise of using the Connectome 2 scanner at 3T to look at these high-resolution ASL data. But there are several opportunities for further improvement, including using some of the avenues that Renzo mentioned in his talk this morning to improve the acquisition, to improve the resolution further, and also to use different readouts to be able to reduce our scan time down from 18 minutes to something that's more clinically feasible. And lastly, we've also acquired, in addition to our single delay data, also ASL data, multiple post-labeling delays, which also allows us to measure arterial transit time in addition to CBF data. And so we're currently working on that data as well to look at how the arterial transit time varies across the cortical ribbon. So with that, I'd like to thank my team. So Jan Kufer and Hannah, who I believe are the ones who largely acquired this data, and Teddy Martin here is the one who did a lot of the data processing that I've shown you today. So thanks also to Susie and Ari for facilitating the acquisition of this data. Thanks again.